Hello. This video will cover the structure of the cell membrane. Remember, as we discussed earlier in class, the function of the cell membrane is to provide support for the cell, act as a barrier to the exterior environment, and aid in movement of the cell. The membrane also allows for the passage of some materials into and out of the cell, and we refer to this as being semi-permeable. Some of you may have a copy of the cell membrane handout that you can follow along with. This video is going to focus on this section of the handout. The first component of the cell membrane we are going to discuss is the phospholipid. The phospholipid is a macromolecule. It is a lipid, and it is depicted here in this yellow square. Notice if we look at the molecular structure of the phospholipid, it has a polar phosphate choline head made up of a phosphate molecule and a choline molecule that are bonded together, and these are attached to a fatty acid tail. There are two fatty acid tails, and these are the nonpolar parts of the phospholipid. The phospholipid is the main component of the cell membrane. Again, each phospholipid contains a polar head that is made up of a phosphate and choline, and it also contains two nonpolar fatty acid tails. All of the nonpolar tails are going to turn towards one another in an attempt to exclude as much water, which is a polar molecule, as possible. The phospholipids are going to come together and form two layers of phospholipids, which will be oriented so that each layer has its hydrophobic fatty acid tails facing each other in order to create a hydrophobic core. The bilayer only allows some substances to diffuse through. These substances must be small and nonpolar, such as oxygen gas, or they may be small and polar. For example, water can sometimes diffuse through, but they cannot cross the bilayer if they are charged, in the case of sodium ions, or if they are large macromolecules such as glucose. Notice that the phospholipid bilayer creates an outer layer and an inner layer. They are not symmetrical layers because each layer serves a different function. The outer layer contains sugar chains and proteins, which are referred to as glycoproteins, as well as sugar chains on some of the phospholipids, referred to as glycolipids. These structures aren't found on the inner layer of the membrane. These glycoproteins and glycolipids are involved in cell recognition and cell-to-cell communication. The outer layer also contains receptor proteins that bind hormones and growth factors as a means of communication. These are also absent in the inner layer. The inner layer specializes in binding elements of the cytoskeleton. Recall that the cytoskeleton serves as structure for the cell and is involved in movement of the cell membrane. The cell membrane is described as a fluid mosaic. This means that it is not stationary, it is not static, but it is moving. It also means that it's made up of multiple components. One of the major components making up the cell membrane, besides the phospholipids, are proteins. Proteins can be broken into two categories. The first category are integral proteins. These are also known as intrinsic proteins. Integral proteins have at least one region embedded within the hydrophobic core of the bilayer. The most typical type of integral proteins are transmembrane proteins, which span the entire bilayer. These can be exemplified here, here, and here. Some integral proteins are associated with one side of the membrane. However, they do not span the entire membrane. For example, this protein structure here is located on the outer surface of the cell membrane. It does extend into the inner part of the hydrophobic core, but it does not extend all the way through to the inner part of the cell membrane. The second category of proteins are peripheral proteins. These are also known as extrinsic proteins. These are proteins that lie on and are secured to the inner or the outer surface of the membrane. Notice this protein is associated with the inner surface of the membrane, and this protein is associated with the outer surface of the membrane. Peripheral proteins do not interact with the hydrophobic core of the bilayer. There is no extension of the protein into the hydrophobic core as there is with integral or intrinsic proteins. The final major component that we'll be discussing in the makeup of the cell membrane are cholesterols. Cholesterols play a role in providing the membrane with the proper amount of fluidity and stability. At high temperatures, they reduce fluidity by restricting the movement of the phospholipids. At low temperatures, they keep the membrane more fluid by preventing the fatty acids from forming a non-fluid gel. For example, when some oils are cooled, like coconut oil, they lose their ability to flow and become a semi-solid gel. It is important to note that plant and bacterial cells do not contain cholesterol in their cell membranes. Discussing the phospholipid bilayer, we mentioned that the outer and inner layer are not symmetrical because there are structures that are only found on the external layer. One of these structures are oligosaccharides or sugar chains. These oligosaccharides or sugar chains will attach to either a protein or a phospholipid. When attached to a phospholipid, they are referred to as a glycolipid. This is a phospholipid that contains a sugar chain on its surface. Glycolipids function as markers for cell recognition. For example, antigens that determine the ABO blood group. Glycolipids also provide a source of energy to the cell. Oligosaccharides also attach to proteins. When they are attached to proteins, this structure is referred to as a glycoprotein. This is a protein that contains a sugar chain on its surface. Glycoproteins will serve many different functions, from structural components to receptors to enzymes. As you review your notes on the cell membrane, pay special attention to the phospholipid and its structure, its role in making up the phospholipid bilayer, the functions of the phospholipid bilayer, the role of cholesterol, the role of glycolipids, and the role of glycoproteins in the cell membrane.